Okay, my name is Travis Tebow. This is Mikayla Streif. We're doing the recorded dramatization for pharmacology on our final project. I will be playing part of the nursing instructor, and Mikayla is the student, and we'll be discussing metabolism. So, first thing we'll be discussing is absorption. The method of delivery of a medication affects how it is absorbed, the onset and length of action, and bioavailability of a drug. During absorption, drugs interact with various receptor molecules to produce a desired physiological effect. Different variables that affect absorption include the nature of the cell membrane, blood flow at administration site, solubility of the drug, pH, molecular weight, drug concentration, and drug form. So Mikayla, do you know the main routes of administration?
Yes, I do. The main routes of administration include oral, buccal, sublingual, intermuscular, and intravenous. The onset and duration of a drug reflect the route of administration and the pharmacokinetics of the drug and properties of the receptor. When considering which route to use, bioavailability needs to be considered. Bioavailability is the percent of medication that is observed. The IV route will produce the most rapid effect as it is instantly in the bloodstream, which results in 100% of bioavailability of the drug as it is not broken down by saliva, the GI tract, or the liver, which avoids the first pass effect.
Good job, Mikayla. I would like to add that times during drug response vary for each route of administration, and dosages for each may be more or less as absorption affects bioavailability. Oral medications are used most often due to their convenience and compliance outside of the clinical or hospital setting. The stomach dissolves contents of oral medications so that when it reaches the intestines, they are able to enter systemic circulation. The next topic we'll discuss is distribution. This is the process of a drug moving throughout the body to the target sites. Distribution requires areas of adequate blood supply to function properly. If a drug does not reach the intended target site in sufficient concentration, a therapeutic effect will not take place. The size, charge, chemical structure, and lipid water solubility of a drug can affect cell transportation systems and diffusion of a drug through barriers such as cell walls. When a drug is small and uncharged, passive diffusion occurs more easily. And passive diffusion is the process in which medications cross a biological barrier. Blood pH influences the amount of drug that is charged and uncharged, and correspondingly results in decreased or increased diffusion. The higher the acidity level, the lower the pH level that can affect ionization. The average pH of the stomach is 2, intestine is 6 to 8, and blood is 7.4. So Mikayla, what information can you add regarding distribution?
I would like to add that drugs combine with plasma proteins or be what we call free drug in the bloodstream. The free drug plus the bond drug produces the drug-protein complex. Free drugs are active drugs that can be dynamic, extending the amount of time the drug is available in the body. Protein binding allows for free circulation in the blood, which allows, when they're unbound, for passive diffusion at target sites, since diffusion of drugs cannot occur when they are bound to proteins because the distribution is affected. Plasma proteins are produced in the liver. For example, albumin is a plasma protein, and the amount of that in the blood can be helpful in determining liver function. Since plasma protein levels can be affected by liver function, nutritional status, aging, and disease, these also need to be considered. For example, patients with poor nutrition, cancer, liver disease, or other infections may have low plasma proteins, causing more free drugs circulating in their blood rather than protein-bound drugs.
Great information, Mikayla. To continue your point, it is noted that due to these drug-protein properties, proteins can help normalize drug concentrations throughout the body and prolong the amount of time a drug is in circulation. Also, when drugs are highly protein-bound, even small changes in the amount of protein-bound drug can result in dramatic changes in the free drug, as is seen with warfarin. Mikayla, do you know why warfarin is an example of this?
In fact, I do, Mr. Travis. Warfarin is a drug that is 98% highly protein-bound, illustrating how small changes, such as other drugs competing for protein binding, can result in dramatic changes in free drugs in circulation. If a patient takes a second highly protein-bound medication in congruence with warfarin, it can alter the effect, resulting in non-therapeutic levels.
Good job. In conclusion for this topic, volume of distribution also is important as it helps determine where a drug is being distributed throughout the body and an understanding of blood levels in relation to drug dose. The next section we will discuss involves metabolism. Metabolism is the process of breaking down and building up substances to maintain bodily function, usually through enzymes. An example includes oral medications metabolized by the liver, demonstrating first-pass metabolism to determine if that medication can be administered orally to have a therapeutic effect. Metabolism determines drug activity, which affects the pharmacokinetics of the drug. Through the metabolism process, the drugs are altered into new molecules called metabolites. The body consists of many different enzymes that cause various chemical reactions. Mikayla?
I recall reading information on this topic. For example, the CYP450 family metabolizes a large number of drugs administered every day. Various factors that can influence metabolism includes age, genetically determined differences, pregnancy, liver disease, time of day, environment, diet, alcohol use, and drug interactions.
Yes, you are really understanding these concepts. There are two different phases of metabolism, that first being phase 1 metabolism, also called non-synthetic reactions. Xenobiotics undergo metabolism primarily in the liver through oxidation, reduction, and hydrolysis reactions, generally improving water solubility and in preparation for further metabolism or excretion. Phase 1 reactions introduce or mask polar groups that, in general, improve water solubility and prepare drugs for further metabolic reactions. During phase 1 reactions, metabolites are rapidly eliminated. Mikayla, can you tell me about the second phase?
Actually, I can, yes. During phase 2 metabolism, also called synthetic or conjugation reactions, right?
Correct.
Metabolites are linked to polar molecules, making them even more water soluble and excretable. During this type of metabolism, drug molecules are metabolized and something is added to the drug to synthesize a new compound. Metabolites are linked or conjugated to highly polar molecules. Conjugation allows such molecules to make metabolites more water soluble, which we learned makes the excretion process by the kidneys easier. The enzymes produced during phase 2 reactions can influence the pattern of drug activity and the duration of the action of drugs.
Very good, Mikayla. And for your reference, I am going to hand you an example of what phase 1 and phase 2 metabolism is in phenobarbital. There you go.
This is great. I really understand it better with this illustration.
Well, I appreciate that. And the last topic we'll be discussing is the cytochrome 450 enzyme system, or referred to here as CYP450, is a family of enzymes found mostly in the liver that involve the biosynthesis and degradation of compounds, chemicals, toxins, and medications. Over 50 CYPs have been identified and are further classified by family, subfamily, and individual genes. Mikayla, can you define a substrate for me?
Why, yes, I can, Mr. Travis. A substrate is the medium in which a chemical reaction takes place, or the reagent in a reaction that provides a surface for absorption. In biochemistry, an enzyme substrate is the substance the enzyme acts upon. When referring to the CYP450 system, substances metabolized by a CYP450 enzyme is a substrate.
Very good.
Thanks.
The next question we will answer is, what are inducers and inhibitors, and how do these affect the CYP450 system? Inducers are those agents, usually food or drugs, which increase the rate of metabolism of a substance, thereby eliminating it from the body more quickly, which can lead to reduced or no efficacy at all. Contrastingly, inhibitors are agents that inhibit the metabolism of a substance, leading to a rise in blood levels and potential toxicity. Single nucleotide polymorphisms, or SNPs, are mutations in proteins that can result in metabolic changes as well. When SNP variations exist in individual CYP enzymes, they are denoted with an asterisk, and can result in increased, decreased, or normal metabolic activity. These may also be referred to as phenotypes, 4 of which may exist. Can you name the 4 phenotypes, Mikayla?
I can. One is the ultra-rapid metabolizers, which you use UM as an abbreviation, extensive metabolizers, which you use EM, intermediate metabolizers, which are IM, or poor metabolizers, which are PM. These alterations in DNA are sometimes associated with population groups, which explain why some of these populations are more or less sensitive to certain drugs.
Very good. Next, we're going to talk about functioning and non-functioning metabolism. The first example of metabolism is when a person with chronic pain uses a fentanyl patch for a sustained slow release of the medication and long-term pain control. In functional metabolism of fentanyl, it undergoes phase 1 metabolism by CYP3A4 and CYP2D6, and phase 2 metabolism by conjugation. Its metabolites are then excreted through the urine. Mikayla, can you think of another example?
I can. Another example of functional metabolism is when a person with hypertension is administered a calcium channel blocker such as amlodipine. With normal metabolism, amlodipine undergoes phase 2 metabolism via the CYP3A4 and phase 2 conjugation and circulates with greater than 93% being protein bound and excreted in the urine. In both instances, therapeutic doses of free drugs circulate, providing the desired therapeutic effect.
That is correct. On the other hand, an example of non-functional or variable metabolism is when a person who is an ultra-rapid metabolizer of the CYP2D6 enzyme is administered codeine, which is a prodrug and substrate of CYP2D6, resulting in toxicity as it is converted to morphine at a faster rate than normal, resulting in an undesired increase in blood concentration. Mikayla, can you give me one last example of non-functional metabolism?
I do recall one other example that we discussed of non-functional metabolism that can result in toxicity when the CYP3A4 substrate, such as colchicine, is combined with a CYP3A4 inhibitor, such as grapefruit juice, erythromycin, or ketoconazole, which can lead to fever, diarrhea, muscle pain, paresthesia, or death. This is because inhibition of the enzyme results in delayed metabolism and results in increased circulation of the drug, which will end in toxicity.
Great job, Mikayla. Well, this class had a great discussion today, and it is apparent you understand these concepts well, which is a good thing because you'll be tested on all of them tomorrow. Happy studying and good luck. You're going to need it.